carbohydrates. Carbohydrates can be of two types, simple carbohydrates and complex carbohydrates. Simple carbohydrates means it contains only carbohydrate unit, whereas complex carbohydrates, they have carbohydrate unit but with lipid or protein, and it can be of three types, glycoproteins, proteoglycans or glycolipids. This we will be discussing in next video, but in this video we will be discussing about simple carbohydrates. Now, simple carbohydrates can be classified as monosaccharides, oligosaccharides or polysaccharides. Mono means a single unit. Means monosaccharides will contain only a single carbohydrate unit. Whereas oligosaccharides, it contains anything between 2 to 10 carbohydrate units. Suppose we are talking about disaccharide. It means it will be made up of 2 monosaccharides. Whereas polysaccharides have 11 or more than 11 carbohydrate units in them. Now, starting with monosaccharides first. Monosaccharides can be classified on the basis of number of carbon atoms or on the basis of functional group. Now, when we are classifying it on the basis of number of carbon atoms, it can be triosis if it has 3 carbons in it. It can be a tetrosis if it has 4 carbons in it. It can be a pentosis if it has 5 carbons in it, and hexosis if it has 6 carbon in it, and so on. And about the functional group, if it has a aldehyde group, it will be classified as aldose sugar, and if it has a ketone group in it, it will be a ketosis. Now, for our understanding, let's take an example first. This is the simplest example we all know, glucose and fructose. They both have 6 carbons in it. Glucose has 6 carbons and fructose has 6 carbons. It means it is a hexose sugar. But this glucose, it has a aldehyde group in it. It means it is a aldose. Whereas this fructose, it has a ketone group in it. It means it is a ketose. Now let's take one another example. This is a ribose. It has 1, 2, 3, 4 and 5 carbons in it. It means it is a pentose sugar, and it has a aldehyde group. It means it is a aldose sugar. Now this was about ribose, but if we want to make it a ketone group, so what will be the name? We'll simply add UL in it. Whenever we want to name a ketone group out of aldehyde group, we simply add UL in it. Like ribose, so ribulose, erythrose, so erythrulose, like that. Now taking some more examples. Now this all are hexose sugars. And when we compare galactose with glucose, it has a difference only on 4th carbon. It means it is a C4 apimers of each other. And when we compare this glucose with mannose, it has difference only at the 2nd carbon. It means it is a C2 apimers of each other. This was all about apimers. But when we compare glucose and fructose, they have everything same except this functional group. It means it is a isomer. And if they are isomers, there can be functional isomer or structural isomer. They have structural formula same, but their functional group is different. That means they are functional isomers. But when we compare this mannose with galactose, they have 2 difference in it. It means it can't be a apimer, because for apimer there should be only difference between 1 carbon atom. Like in galactose and glucose, there was difference only on the 4th position. But in mannose and galactose, there is difference on 2nd and 4th carbon both. That means it is a isomer. But their functional group is same. They both have aldehyde in it. It means it is not a functional isomer. It is a structural isomer. Now about the enantiomers. What is an enantiomer? So this is an image of glucose, but they are mirror images of each other. As you can see, this is on right side, this is on left side, this is on left side, and this is on right side. They both are on right side, whereas they both are on left side. It means they are totally a mirror image of each other. And whenever any molecule is mirror image of each other, we call it as enantiomer. And we can classify it as D-glucose and L-glucose. Now, on what basis we are classifying it as D-glucose or L-glucose? So, we'll see this OH, the second last one. When it is on right side, it is called D-glucose. But when it is on left side, it is called L-glucose. The same way any molecule is named. Now we know this is a glucose molecule. But if we join it like this, forming a structure like this or a structure like this, it is called Fischer projection. Now what's the difference in these two structures? So you can see this OH is on left side, whereas this OH is on right side. When it is on right side, it is called alpha glucose. But if it is on left side, it is called beta glucose. But we can also form a structure like this, a ring-like structure. And when it is a ring-like structure, we called it as Haworth projection. And it can be also of beta type and alpha type. So for alpha type, we have to see the first carbon. If the OH is on the lower side, it means it is a alpha glucose. But if the OH is above the plane on the first carbon, it is a beta glucose. Now we are done with monosaccharides, starting with oligosaccharides. The most important one is disaccharide. And under disaccharides, there are 5 important disaccharides: maltose, isomaltose, trehalose, sucrose, and lactose. Now about the sugars. The maltose, isomaltose, and trehalose, they all have glucose plus glucose combination, whereas sucrose have glucose plus fructose and lactose has galactose plus glucose. Now the link between them is alpha 1 4 in maltose, means glucose plus glucose is attached at 1 4 linkage, that is alpha 1 4 linkage, whereas isomaltose, it is attached at alpha 1 6 linkage. And the trehalose, the linkage between them is alpha 1 1. And the sucrose, it has linkage alpha 1 2. But the lactose has beta 1 4. Now we know that our human digestive enzymes can't break beta bonds like of cellulose. Cellulose also have beta 1 4 linkage. But lactase enzyme, the enzyme which converts lactose into galactose and glucose, is the only exception which can break beta bonds. But if there is any defect in this lactase enzyme, it will lead to lactose intolerance. Now, what is this lactose intolerance? For that, we should know the normal mechanism. So, any monosaccharide is absorbed via this intestinal villi. But lactose is a disaccharide, and disaccharides can't be absorbed directly. It need to be break down into galactose plus glucose, and it will be done by lactase. But in some patients, this lactase is absent, leading to lactose intolerance. Lactose intolerance is a digestion defect, and as lactose is a disaccharide, it will stay in lumen. And as disaccharides are osmotically active, it will attract water, causing diarrhea. And this disaccharide will reach the colon, and we know that colon has microorganisms in it which will convert lactose into methane and hydrogen. And this hydrogen and methane gases will be accumulated in colon. And because of that, we can do hydrogen and methane breath test of the patient to know if the patient is lactose intolerant. And because of these gases, there will be flatulence, frothy stools, and also bloating will be seen. And this microorganism will also act on lactose to form acids. And this acid is responsible for acidic pH, which is responsible for perianal excoriation. And this acidic pH is observed in stools also, which can be an investigation of choice. But hydrogen or methane breath test is normally investigation of choice, but stools are investigation of choice in neonates. Now we are done with oligosaccharides, starting with polysaccharides now. Polysaccharides can be of two types, homopolysaccharides and heteropolysaccharides. Homo means one, whereas hetero means two. Now about the homopolysaccharides. It is further classified into two, the structural one and functional. Function means storage, and when we want to store anything, it should be branched, but for structural it is unbranched. Functional are starch and glycogen. We know starch in plants and glycogen in animals. Whereas in structural, we have cellulose, then we have chitin, and also we have inulin. Now starting with cellulose. Cellulose is a component of plant cell wall and it is made up of multiple glucose. And this multiple glucose is linked with beta 1,4 linkage. Now about the chitin. It is present as an exoskeleton this in the scales of snakes and cockroaches, and it is made up of NAG, that is N-acetyl glucosamine. Now about the inulin. This inulin, it is used to measure glomerular filtration rate and it is present in plant tubes and it is made up of fructose, and it is very important that it is made up of fructose. Now about the starch. The starch has two components. One is amylopectin and one is amylose. You can remember as pec means branched and lose means unbranched. Now if it is branched, it will have two linkages. One along the straight chain, that is alpha 1,4 linkage. And when it is branched, at every point of branching, it will be alpha 1,6 branching. And as it is branched, it will melt at high temperature, that is 50 degree, and it is present as solid. But amylose has only one linkage along the straight chain, that is alpha 1,4, as it is unbranched, and it will melt at very early temperature, that is just 15 degree, and it will be present at liquid form at room temperature. Now about the glycogen. Glycogen is present in liver and muscles and it is highly branched, and it is a spiral molecule which has glycogenin at the center of it. And this glycogenin will attach glucose residues to it, 11 to 13 glucose residues. It can be direct connection or indirect. If it is direct, there will be alpha 1,4 linkage, and if it is indirect, then it will be alpha 1,6 linkage. And this entire structure is arranged in 12 concentric rings to enable compactness. And more number of branch structures will be present at the center. Towards the center, more branching will be seen as compared to the periphery. Thanks for watching. Don't forget to like and subscribe and share with your friends.